Hi guys, this is Ms. Romani, and today we're going to be learning about genetic testing. So let's dive in. Basically, genetic testing is a type of biotechnology that aims to identify people who currently have a genetic disorder or even people who may be at risk of developing a disease that has a genetic link or who is at risk of passing a genetic trait to their offspring, but they themselves are healthy. So there's a variety of different types of genetic testing, and we're going to go through a lot of these different types today with examples, and hopefully you're going to be able to know a little bit more about genetic testing by the end of this lesson. So to start, let's take a look at what we may be looking for when we're looking at genetic testing, because there's three approaches to genetic testing, three different places where we can look for genetic abnormalities. So genetic testing may actually look at the chromosomes through a process called karyotyping that you may have learned about in grade 11 biology. And karyotyping looks at the number and the shapes of the chromosomes, and the chromosomes contain the DNA. So we can also take a look at the DNA. And these are DNA or molecular or gene tests. And molecular or gene tests, sometimes also called genetic tests, even though all of these are genetic tests, but molecular or gene tests actually look for specific mutations or specific variants of genes. So they're looking for the gene itself. Or we can take a look at the products of the genes, which are the proteins. And we do this through a process called newborn screening. And this is an indirect type of genetic test because we're not actually taking a look at the DNA or even the chromosomes. We're just taking a look at the product of DNA itself, seeing if there is the right protein that was made or whether there's extra protein or the wrong type of protein or if there's missing proteins. So when I teach about genetics, I often like to use the analogy of recipe books. A gene is the instructions in our cells to make proteins, in the same way that a recipe would be the instructions in our kitchen to make, let's say, cookies. So if the gene is the recipe, then the protein would be the cookies. What would be the chromosomes? The chromosomes would be the recipe book. And we don't have just one recipe book. We would have several recipe books. We actually have 46 recipe books. And so these recipe books contain the pages inside of them, which is the DNA, which have the letters, which is the genetic code. And in some of those pages, the instructions for making cookies. The analogy here would be like this. Instead of looking for just the recipe, we can look at our library of recipe books, and you can count the recipe books. You can make sure that there are no missing recipe books, that there are no extra recipe books, that maybe some of them are not missing entire sections of the recipe book. When we take a look at the DNA or molecular or gene test, it's like we're looking at the recipe itself and we're checking for typos. We're checking for mistakes. And then the last way we can do that is instead of looking at the recipe books or the recipes, we just take a look at the cookies. Did we make the right cookies? Did we make some of the cookies with the wrong ingredients? That tells us that maybe we made a mistake in the recipe. Some cookies are missing. Maybe the cookie is missing because the recipe was so wrong that the cookie couldn't be made at all. That's basically the analogy, right? So we can look for the books, the chromosomes, we can look for the recipes, the DNA, or we can look for the cookies, the proteins. So let's take a look first about chromosomes. So chromosomes can be examined through a process called karyotyping. And a karyotype is essentially just a picture of all the chromosomes that are found during metaphase, during mitosis at metaphase, and organized in a way that organizes the chromosomes by their pairs, starting from the biggest chromosomes all the way to the smallest chromosomes and putting the sex chromosomes at the end. And the reason we do this is because by organizing the chromosomes this way, it's much easier to tell if there's any missing, any extra, or any that are misshapen or missing pieces. Let's say, for example, if we go back to our recipe book analogy, if I give you a big pile of recipe books and I say, quick, tell me what's wrong with these books, you wouldn't be able to do that very quickly. But if instead I showed you to my library where there's a shelf and all my recipe books are organized on my shelves by size and by type, paired up properly, then you very quickly would be able to tell me if all my books are there, if there's any missing, if there's any extra, and which of them might have some problems. And so that's basically how a karyotype works. It's just a way of organizing our chromosomes so that we can better diagnose chromosomal abnormalities. And an example of a chromosomal abnormality is something like Down syndrome. Now, karyotyping can be done either after birth or before birth, which is called prenatally. And oftentimes karyotypes are done prenatally, but they can also be done after a baby is born. If there's an indication that there's something wrong with the baby and they might have a chromosomal abnormality. And sometimes for some chromosomal abnormalities, you might not even detect them until a child fails to reach puberty properly, or if they start to develop certain symptoms once they hit adolescence. These are usually the chromosomal abnormalities that relate to the sex chromosomes. And so karyotyping is not always done before birth, which is called prenatally, but it can be, and it often is. So when we take a look at this karyotype, right away I can tell two things about it. The first one is that this child has Down syndrome. And the reason we can tell is because there's an extra of chromosome 21. And the second thing I can tell about this child is that it's a girl, because she has two X chromosomes. And if the baby was a boy, it would have an X chromosome and a Y chromosome. So they would have one big chromosome and a tiny little chromosome called the Y chromosome. So we call chromosomes that are not the X chromosomes or the Y chromosomes, we call those autosomes. So all the chromosomes from chromosome number 1 to chromosome number 22 are autosomal chromosomes. And notice that they're in pairs, right? Because we get one of each type from each parent. So we get one of each type from our mom and one of each type from our dad. And then the X and Y chromosomes are simply called the sex chromosomes. So those are the two types. So I mentioned that chromosomal abnormalities can be tested prenatally, and that is actually true for other genetic disorders as well. But the key to testing prenatally is obtaining cells from the baby before it's even born. And there's a couple of ways that this can be done. One of them is through a process called amniocentesis. Now, amniocentesis requires the insertion of a needle into the placenta in order to extract some of the amniotic fluid. And the reason that this is done is because once the baby's inside the womb, the baby will shed some of its skin cells, and so those skin cells will be found within the amniotic fluid itself. Now, amniocentesis can only be safely performed within a narrow range of time while the baby is in utero. And so once the amniotic fluid is removed, the amniotic fluid can be centrifuged so that the fetal cells can be separated. They need to be cultured in a petri dish so that they can grow and divide, so that we have enough cells so that then a karyotype can be performed. And not just a karyotype, they can also do genetic testing on those cells also. This process takes a few weeks to complete due to the fact that the cells need to be cultured and so on. So amniocentesis is a test that cannot be performed too early during fetal development, and also takes a few weeks for the results to come in after the test itself is done. Now, another type of prenatal genetic testing is via chorionic villus sampling. And so chorionic villus sampling can be done earlier than amniocentesis because it doesn't require the presence of enough amniotic fluid, because the cells from the fetus are not isolated from the amniotic fluid. They're actually obtained directly by taking a sample of the placenta, and the placenta is actually made up of fetal cells. There is a layer of cells in the placenta called the chorionic villi, which are part of the placenta, and those are extracted through suction. So in order for those cells to be extracted, a probe is inserted through the cervix and into the uterus in order to extract some of those cells. Now, because those cells are extracted directly, they don't need to be isolated, they don't need to be cultured, they can be tested right away. So this particular type of test actually takes only a few hours to complete. So this is a much more efficient prenatal test. However, it does carry a higher risk of miscarriage than an amniocentesis does. As a woman ages, doctors will be more likely to recommend prenatal testing for chromosomal abnormalities, especially for women that are pregnant after the age of 35. And the reason for this is because as a woman ages, the risk that she will conceive a child with a chromosomal abnormality increases significantly, especially after the age of 40. But the risk is higher about the age of 35 as well. So, for example, see this graph right here, and you can see two lines. The first line in blue is showing the risk of chromosomal abnormalities or the prevalence of how often they occur for every 1,000 births for women based on their age. So at the age of 20 to about age of 25, there is no significant difference in the prevalence of chromosomal abnormalities. But after that point, it starts to go up slightly. And then after age 35, it really starts to go up exponentially. And that is because a woman is born with every egg she will ever produce in her lifetime. And so by the time that a woman hits the age of 35, her eggs are 35 years old. A 25-year-old woman has 25-year-old eggs. So there is a difference in the viability of the eggs as more time passes. So, for example, if we look at some data directly, a woman who conceived at age 40 is more than 10 times more likely to conceive a baby with Down syndrome versus a woman who is 25 years old, which is the reason why prenatal testing is recommended for women over the age of 35. And so one ethical concern with this type of genetic testing is the increased incidence of termination or pregnancy terminations as a result of the tests, especially when it's done prenatally. This data from Australia shows the incidence of Down syndrome abortion rates compared to the incidence of Down syndrome pregnancies that are diagnosed prenatally. As more and more tests are done, more and more terminations happen for babies with Down syndrome. Okay, so let's take a look at how do we look at the genes themselves, molecular or gene tests. And molecular or gene tests are basically tests that are done on the DNA itself. They test for either very specific mutations or gene variants, which we call alleles. Now, why do I make the distinction? A mutation is a mistake. So a mutation will lead to a genetic disorder, whereas a gene variant is just a different code for the gene. And sometimes gene variants can lead to some genetic disorders, but sometimes gene variants can lead to things like the inability to taste cilantro. So genetic tests can test for both either directly a genetic mutation, which causes a genetic disorder, but also variations of genes, so different alleles for genes, so that we can determine whether somebody has the gene for blonde hair as opposed to red hair. And that's neither one of those are mutations per se, they're just variations of the gene. So there are several types of gene tests that can be performed. And the one that probably comes to mind right away when you think about genetic testing is diagnostic gene testing. So this is done to identify or to rule out a specific genetic condition on a patient. So somebody thinks they might have a genetic disorder, and this test can be done to right away figure out if they actually do have the genetic disorder, or to rule out and say, hey, good news, you don't have it. So, for example, is the test for cystic fibrosis. Cystic fibrosis is the most common fatal genetic disease affecting Canadian children and young adults. And unfortunately, there is no cure for it. So cystic fibrosis causes a lot of different effects on the body. Mainly, it affects the digestive system and the lungs. And the disease is actually caused by a single gene, a single mutation in a single gene that causes the formation of a non-functioning protein. And the protein that is supposed to be made is an ion channel. In children with cystic fibrosis, the ion channel that is formed is faulty, it doesn't work, and that ion channel is supposed to control the movement of both salt and water in and out of body cells. So symptoms of the disease basically are as a result of this inability to control the osmotic gradient of the water in and out of the cells. It causes really, really thick mucus to form. Symptoms of the disease will actually present itself to be serious enough that parents who have an undiagnosed child will seek medical treatment by the age of 2 years old. Now we can confirm that diagnosis with an actual genetic test that looks for the actual cystic fibrosis mutation in DNA. So that's diagnostic testing. Another type of testing that can be done is carrier testing. Carrier testing is used to identify not people who have a genetic disorder, because they are healthy actually, is just to identify people who have one copy of a gene that codes for a recessive genetic disorder. And we call those people carriers. So these are basically used to identify heterozygous for a recessive genetic condition. And this type of testing is basically offered usually to individuals who have a family history of a genetic disorder, or maybe have a particular ethnic background that makes them more likely to have a family history of a particular genetic disorder. So for example, let's take a look at Tay-Sachs disease. Tay-Sachs is a disease that results in the destruction of nerve cells in both the brain and the spinal cord. And people of certain ethnicities are more likely to be carriers for Tay-Sachs disease because it runs in those family groupings. In particular, people of a very specific Jewish heritage, people of French Canadian heritage, and also people with Irish heritage tend to have a higher risk of carrying the Tay-Sachs gene. So somebody could be healthy, but they could be what are called carriers. So let's say you have a man and a woman and they meet and they happen to maybe be from one of these ethnic groups, or they have people in their own families who have Tay-Sachs disease. So they are concerned that they actually may be carriers. Even though they are healthy themselves, they have no disease symptoms, they could have within their genes one copy of the recessive gene for the disease. And so let's say, for example, they get tested, and if they both happen to be carriers, they would be heterozygous, so they each would have the dominant allele and the recessive allele for the disease. And so the way it would work is that they would be able to pass on both of the recessive alleles to 1/4 or 25% of their children, one of their dominant and one recessive allele to half of their children, 50% of their children, and both of their dominant alleles to the other 1/4 or 25% of their children. In a situation where two carriers marry each other and have babies, they have a 25% chance of having a child with the recessive genetic condition. Okay, so now let's talk about predictive genetic testing. Predictive genetic testing is used to identify mutations that don't necessarily guarantee that you will develop some sort of disorder, but it just increases the chances that that disorder might be present in your future because there's a genetic component to the disorder itself. So, for example, certain types of cancer, in particular breast cancer, have been linked to, in this particular case, two genes, the BRCA gene. The BRCA1 and the BRCA2 genes are the two genes that are usually mislabeled as the breast cancer genes. So let's say you have a history of breast cancer in your family. Maybe you had a mother or an aunt or a grandmother that had breast cancer, and you're concerned that you might be a likely candidate to have inherited the breast cancer gene or one of the variants of the BRCA1 or BRCA2 genes. And so you go and get genetically tested for that. What that means is that if you actually have, let's say, the BRCA1 gene variant, your chances of having cancer or breast cancer will increase from 12%, which is the general population rate, to 55 to 65%. So it is a much higher chance that you will develop breast cancer, or more than 50% chance. The BRCA2 gene gives you a 45% risk of breast cancer as opposed to 12%. But the BRCA genes actually are associated with much more than just breast cancer. And so mutations in those genes increase your incidences of ovarian cancer, and for males, male breast cancer and prostate cancer. And then for both men and women, melanoma, which is skin cancer, as well as pancreatic cancer. So there's a few other cancers that are associated with those two genes. The reason why people sometimes get tested for these genes is so that they can make maybe decisions about their medical care or about their lifestyle choices if they are found to have the presence of those gene variations. For example, if you find out that you have either the BRCA1 or BRCA2 gene, or if you're unlucky, both, then perhaps you might get your skin tested more often. You go to the dermatology very often and check for skin cancer on a regular basis. You would definitely avoid the sun and put on tons of sunblock. You would have regular checks with your doctor. If you're male, you would get your prostate checked regularly. And the other thing that you might also do, some women have done this, they are opting to having their breasts removed and then they do reconstructive surgery soon after. But this is a way of avoiding the breast cancer, which is obviously a much higher risk for them. Famously, Angelina Jolie actually had a double mastectomy done because she tested positive for the BRCA gene, one of them. These types of tests are predictive tests, though. They don't guarantee that you'll get a particular disorder. They just increase your chance of getting a particular disorder. And other disorders are also being linked to some genetic component, like, for example, Alzheimer's disease and type 2 diabetes might have a genetic component, as well as some other ones like heart disease and so on. So predictive genetic testing is different than diagnostic testing because even though they are looking for a very specific mutation, it doesn't necessarily mean that you will for sure have or already have the genetic disorder. Another type of genetic testing that can be done when you're still healthy and even though you don't have the disorder is called pre-symptomatic genetic testing. And this one can determine whether or not you will develop a hereditary genetic disorder in the future, even though you don't have any signs or symptoms of it yet. So unlike predictive genetic testing, where you don't know for sure whether or not you'll develop the disorder, with pre-symptomatic genetic testing, you will develop the genetic disorder if you test positive for the gene mutation, but you don't have symptoms yet. And usually this is done for genetic disorders for which symptoms develop later on in life. So let me give you an example. One of the most famous examples for pre-symptomatic genetic testing is for Huntington's disease. Huntington's disease is a progressive brain disorder that causes uncontrolled movements, emotional problems, changes in personality, loss of cognition. This is basically loss of thinking ability. And it is caused by a 3-letter repeat in the genetic code that codes for a brain protein. So these repeats cause the protein to become faulty. And the more repeats a person has, the worse the protein is and the worse the disease gets and the faster it progresses. And so although you are born with a genetic mutation, symptoms of Huntington's disease don't usually start until middle age. And because it is progressive, it gets worse over time. So as the disease progresses, body movements become more erratic or shaky, and affected individuals will actually start having trouble walking, speaking, swallowing. Patients usually live about 15 to 20 years after the signs and symptoms start. And symptoms of Huntington's disease don't appear until after people already had their children. So it is often passed on to future generations. And it is inherited as an autosomal dominant disorder, which means that if a parent develops symptoms for the disease, even though that parent very likely just has a single copy of that affected gene, that single copy leads them to have the genetic disorder, and it also means that their children have a 50% chance of also inheriting the genetic disorder themselves. So oftentimes when people discover that one of their parents has one of these late onset genetic disorders, they might go get genetically tested for these genetic disorders, even though themselves they don't have any symptoms. It's pre-symptomatic testing that is done in order to prepare for the future. In the case of Huntington's disease, there is no cure and there is no treatment, so there's actually nothing that can be done. But there might be some diseases for which some type of preparation or testing or precaution can be beneficial. So I'm going to stop the recording here, and the next recording you're going to see how those at-home testing kits work.